IB Bio Cell Division Meiosis Part II will start with a quick overview of meiosis, then focus on the events of meiosis that result in genetic variation among gametes, variation that ultimately provides the variation within a species. The essential idea is alleles segregate during meiosis, allowing new combinations to be formed by the fusion of gametes. This movie has some higher level syllabus statements, but all of the concepts in this movie are appropriate to both SL and HL students. Meiosis as a reduction division serves to reduce the chromosome number in gametes. Then, when gametes fuse upon fertilization, the diploid condition is restored. And meiosis serves to provide genetic variety from the segregation of alleles, from the exchange of DNA between non-sister chromatids, otherwise known as crossover, and lastly, independent assortment. This movie will examine carefully all three of these events. This movie will also explain how sexual reproduction promotes variation in a species. This graph is an important way to view meiosis. Remember, chromosomes replicate in interphase before meiosis. Then, through two cell divisions, meiosis I and meiosis II, gametes are produced with half the DNA content per cell, half the number of chromosomes. They're haploid. Keep in mind that the two cells formed at the end of meiosis I are haploid, but because all the chromosomes in the cells at this stage are still two chromatid chromosomes, the DNA content per cell is the same as the original cell. Homologous pairs of chromosomes, seen here, have originated from fertilization, with one of each pair arriving from the sperm and the other of the pair from the egg. Homologous chromosomes carry alleles for the same gene, and those alleles have the same locus, the same location on the chromosome. Homologous chromosomes pair up during meiosis I. Alleles, seen here and here, are alternative codes, alternative base sequences for the same gene, and they share the same location on the chromosome, the same locus. We inherit one allele from the father in the sperm and the other allele from our mother in the egg. Remember that we are looking at two chromatid chromosomes here appearing as X-shaped structures following replication, and they're visible to the light microscope during prophase of meiosis I. Remember that sister chromatids are initially genetically identical, capital A here and here, lowercase a here and here. However, crossover, which is a break and swapping of DNA between these two non-sister chromatids, changes that, where the allele here is found here and vice versa, such that non-sister chromatids are no longer identical. Here are the two relevant IB syllabus statements that I presented more thoroughly in Meiosis Part I, with the exception of crossing over and the random orientation of homologous pairs. Here are the two statements. Outline the stages of meiosis clearly, stating the events of each stage. Mention should be made of homologous pairs of chromosomes, two divisions, diploid and haploid cells, and the outcome of meiosis. The second statement, describe the behavior of chromosomes in the phases of meiosis, including crossover, condensation, and the random orientation of homologous pairs, meiosis I metaphase. To understand both crossover and independent assortment, seen here as the random orientation of homologous pairs, you must focus on the pairing of homologs in prophase of meiosis I and their arrangement at the cell equator during metaphase of meiosis I. This movie will review the stages of meiosis, then dig into crossing over and independent assortment. Crossing over happens during prophase I of meiosis, when homologous chromosomes join together and intertwine in a structure known as a tetrad. Regions of overlap, seen here and here and here, are visible to a microscope, and they're known as chiasmata, which is plural, and this is where pieces of non-sister chromatids are breaking and exchanging places. While the notion that pieces of DNA would break and exchange places with other pieces of DNA is contrary to intuition, crossover appears to be enzyme-driven, and the resulting genetic variety has been a powerful selection force over evolutionary time. Now, in metaphase I of meiosis, the homologous pairs move to the cell equator. Crossing over has already happened. Sister chromatids are no longer genetically identical, and the arrangement of one pair, up or down, is random with respect to the arrangement up or down in a second pair or third pair, etc. In anaphase I of meiosis, homologous pairs are separated by one microtubule attached to each chromosome. In telophase I of meiosis, the nuclei reform. Each cell has half the number of chromosomes. The original cell had 4. Each cell here has 2. Each cell here is haploid. In meiosis II, sister chromatids are pulled apart in events that appear like mitosis. I will not review meiosis II in this movie. So here is the looming IB syllabus statement that forms the backbone of this movie, Meiosis Part II. Explain how meiosis promotes genetic variety in gametes through crossing over in prophase I and the random orientation of homologous pairs in metaphase I. When homologs have different alleles, as you can see here, and those alleles separate during meiosis I, the resulting cells are genetically different. You can see this here. One set of gametes has this allele, and the other set of gametes has this allele. I know this doesn't seem like a lot of variation, but be patient. The alleles separate into different cells during meiosis. Mendel, an Austrian monk working with pea plants in the late 1860s, was unaware of chromosomes and meiosis, but he knew from his data that each person carried 2 codes for the same gene, and that these codes, now known as alleles, segregated into different gametes during sexual reproduction. So here we can see how crossover happens. Homologous pairs come together as tetrads during prophase I of meiosis. They intertwine and non-sister regions break and exchange places. You can see crossover here as indicated by the color change of these non-sister chromatids. If you imagine different genes along the length of the chromatid, crossover creates new combinations of alleles on the chromatid. The non-sister allele for a gene is now here, and it's now linked together with alleles of other genes that it hadn't been before crossover. The other allele is now here, newly linked to alleles of other genes that it hadn't been linked to prior to crossover. I'll explain crossover a little bit better in the slides to come. Be patient. So here are the 2 HL-IB syllabus statements relating to crossover, and they will guide the next section of this movie. Explain how crossing over between non-sister chromatids of a homologous pair in prophase I can result in an exchange of alleles. Crossing over should be understood as the exchange of genetic material between non-sister chromatids, resulting in new combinations of alleles on the chromosomes of haploid cells. When homologous pairs come together during prophase I, the structure is known as a tetrad, referring to the 4 chromatids. A chiasma is a region of overlap of non-sister chromatids, a region where crossover occurs. Study these terms so that you understand their meaning. Here's another slide to strengthen your understanding of homologous pairing in prophase I, seen here. Here is the tetrad with chiasmata here and here. This is where crossover occurs between non-sister chromatids. You can see the effect of crossover here and here. Crossover has resulted in new combinations of alleles along the length of the chromatids. In this slide, maybe you can better see the genetic variety that occurs due to crossover. Here's the tetrad. Initially, sister chromatids are genetically identical. Then crossover occurs at the chiasma. The result can be seen clearly below. These 2 chromatids are now genetically different with respect to the 2 genes F and A, and these 2 sister chromatids are now also genetically different. Due to crossover, there are now 4 different genetic combinations. 1, allele capital F with allele capital A. Capital F with allele lowercase a. Allele lowercase f with capital A. and lowercase f with allele lowercase a. Each of the 4 chromatids will end up in a different gamete. Without crossover, there would only be 2 different gamete types, not 4. Homologous pairs align together in metaphase I of meiosis. Notice that crossover has already happened in the first pair. Crossover happens during prophase of meiosis I. Here is a homologous pair, again following genes F and A. No crossover has occurred. There are only 2 genetic types in the gamete seen here. Hopefully you can see how I have abbreviated the 2 meiotic divisions. Think carefully about why this slide makes sense with respect to meiosis. But in this slide, we can see the same homologous pair and notice that crossover has occurred because there are 4 genetic types of haploid gametes. Now you may ask, how do I know that crossover has occurred? It's because sister chromatids are a function of DNA replication, thus they begin their existence genetically identical. And right here, we can see that these 2 sister chromatids and these 2 are not genetically identical. Crossover must have occurred, pieces of DNA swapped places, so that the alleles here are different than what they had been before crossover. Remember that crossover occurs between non-sister chromatids. Crossover between sister chromatids would be meaningless because sister chromatids, before crossover, are genetically identical. With respect to crossover, this slide is identical to the last slide. Due to crossover, you can see the 4 different gametes here at the end of meiosis. But in this slide, I've added another chromosome pair to the metaphase I event, just to remind you that during metaphase I of meiosis, many homologous pairs are aligned at the cell equator. When we look at independent assortment, we'll be looking at the way in which different homologous pairs line up at the cell equator to understand the genetic variety that comes from independent assortment. When we're looking at the genetic variety that arises from crossover, we really only need to focus on one pair, as I have with the F and A genes in these slides. Here's another higher level IB syllabus statement. Draw diagrams to show chiasmata formed by crossing over. Once again, here is our tetrad during prophase I of meiosis. Here is a chiasma, a region of crossover. The 4 chromatids, 1, 2, 3, 4, including the recombinant chromatids, will segregate through meiosis into different gametes. This is an actual micrograph of a tetrad showing chiasmata. Here is another micrograph showing a tetrad with chiasmata. These are regions of crossover. Homologous pairing during prophase I of meiosis results in homologs that intertwine. Non-sister chromatids overlap and exchange segments of DNA. In this diagram, the centromere is shown. Remember, this is where the microtubule will attach as it moves the homologs apart during anaphase I of meiosis. Before I move on to independent assortment, I would like to change the view of crossover that I've shown you up to this point. Hopefully this image of a homologous pair is clear and you know that crossover has occurred. How do you know that crossover has occurred? Is because sister chromatids are no longer identical. If this view of crossover makes sense to you, let me show you the next slide. In this image, I've turned the chromosomes 90 degrees. On the left, the homologous pair is shown with no crossover having occurred. There are 4 chromatids. Each chromosome has 2 chromatids. This is the centromere. Notice that there are only 2 different genetic chromatids. There's been no crossover. Thus, among the gametes, there are only 2 genetic types. On the right, I've displayed crossover. The 4 chromatids can be seen. 1, 2, 3, 4, following each chromatid to a gamete to see 4 genetically different gamete types due to crossover. When we study genetics and we look at linked genes, as we are here, you must be able to use this sideways orientation of chromosomes to display genetic crosses involving linked genes. I've introduced this slide to foreshadow what's coming. Study it and ask questions if you're not sure. One last note on crossover. When following two genes, as I have been doing here with the F and A genes, the frequency of crossover between them depends on their respective distance along the chromosome. If the genes are far apart, then crossover will be more frequent than if they are close together. Now you can see this here with genes B, CN, and VG. Crossover frequency between B and VG is higher at 17% than it is between B and CN or CN and VG at 9.5% each. We can use crossover frequency to map the locations of genes on chromosomes. So I've come back to this IB syllabus statement to examine the genetic variety that originates from the random orientation of homologous pairs in metaphase I. This random orientation of chromosomes and the subsequent movement of those chromosomes to gametes is also known as independent assortment, Mendel's law of independent assortment. I'd also like to briefly state the importance of independent assortment and crossing over in terms of evolution, state that evolution of a species occurs when natural selection selects the most fit among varied forms. In this image, we're following two genes again, gene A and B, but they're not on the same chromosome pair. They are unlinked. They're on different chromosome pairs, and thus, when studying independent assortment, we must view no less than two pairs of chromosomes during metaphase I of meiosis. Notice this particular orientation of the two pairs, and imagine the separation of the homologs through the meiotic divisions. You can see two gamete types that result, capital A with capital B and lowercase a with lowercase b. But could this orientation of the homologs have been different? Sure. In fact, each homologous pair could be arranged in either of two ways, just like we see here or here, where we see the arrangement of this pair different with respect to this pair as compared to the last slide. And here we can see two genetic types that result from this arrangement. Now you might be saying, yes, but I'm only looking at two different genetic types with each orientation, two on the last slide and two on this slide. But keep in mind that millions upon millions of gametes are formed, so each orientation appears in meiosis, resulting in all four genetic types among the millions of gametes. And if you're saying, okay, but only four genetic types, what's the big deal on genetic variety in meiosis? Imagine all 23 pairs for humans aligned during metaphase I of meiosis, each pair randomly oriented in one of two possible positions, and millions upon millions of gametes being formed. The possible combinations are huge. In fact, it's 2 to the 23rd power. In this slide, we can see both orientations that I showed you separately over two slides. They're together here on one slide. The four possible genetic types of gametes are seen here. Independent assortment is defined as the alleles on one chromosome pair assort to gametes independently of the alleles on a second chromosome pair. I know that sounds confusing, so one more time. The alleles on one chromosome pair assort to gametes independently of the alleles on a second chromosome pair. Allele capital A can be with allele capital B, or allele capital A can be with lowercase b, etc. And remember, in humans, 23 pairs are aligned during metaphase I, each pair randomly in one orientation or the other. When millions and millions of gametes are produced, the number of different gametes from independent assortment alone is more than 10 million, 2 to the 23rd power. Here is another image of two possible arrangements when following two genes on just two different chromosome pairs. Notice the four genetic types among the gametes. Here's the definition of independent assortment. And if you've understood everything I've presented so far, you can see the two possible orientations of the homologous pairs here. And if you can imagine two meiotic divisions, you can see the genetic variety in this simple example. In case you're not sure, once again, here are the two orientations. They both occur when making millions of gametes, and we see the four genetic types of gametes on the right following through two meiotic divisions. Notice the genotype given here as capital A lowercase a, capital B lowercase b. The letters are symbols of the alleles. Notice 2 alleles for each gene, and that the alleles are different. Capital A and lowercase a for the first gene, capital B, lowercase b for the second gene. And compare these genotypes to the gametes. Each gamete only has 1 letter for each gene, not 2. Gametes are haploid. They do not have homologous pairs. Gametes have only 1 allele per gene. So here's the IB syllabus statement to which you need to be able to respond. Explain how meiosis promotes genetic variety in gametes through crossing over in Prophase I and random orientation of chromosomes in metaphase I. Hopefully, you have good control of this statement. Here is a summary of the genetic variety that arises from sexual reproduction. Meiosis provides genetic variation among gametes through the segregation of different alleles, independent assortment of alleles, the random orientation of homologous pairs at the metaphase plate of meiosis I, and crossing over. Random fertilization is an event that occurs when 2 individuals engage in sex, and this happens after meiosis. All 4 of these events provide the genetic variety that we see among individuals of a species. Once again, you can stop the movie and read through this slide on your own. With the last 2 slides of this movie, I would like to remind you that each meiotic event in males results in 4 functioning gametes, 4 functioning sperm cells, but meiosis in females results in 1 large gamete, the egg cell, and 3 non-functioning polar bodies. In this image, we can follow meiosis through 2 meiotic divisions to result in gametes. In spermatogenesis, 4 functioning gametes are formed per meiotic event, while in oogenesis, only 1 large egg cell in females is formed per meiotic event. In females, 3 non-functioning polar bodies are also formed. In this image, we can see the size difference between the egg cell, seen here, and numerous sperm cells, seen here. And that brings us to the end of IB Bio, Cell Division, Meiosis Part II. Within the first 2 movies of meiosis, especially this one, I've introduced concepts that will be very important when we study genetics and mammalian reproduction.